Mic check.
Mic check.
Hello, everyone. Welcome to Antibuddies. This is our 20th journal club. And here's the Antibuddies team. I hope you all can see on the Zoom window. And we have a guest with us, Dr. Danyivid Olivares-Villagomez. Before we start with the presentation, let me tell you a little bit about Danyivid. He is an assistant professor at the Vanderbilt University Medical Center, and his lab primarily focuses on mucosal immunology. When we asked Danyivid about his other passions apart from research, he's told us that he's an avid fiction reader and writer. He has written more than 50 short stories, and he just published a book in the magic realism genre titled Claudia. Welcome to the Journal Club, Danyivid.
Thank you very much for the invitation and the opportunity to be here to present this paper that we published last year, that it took a couple of years to work in, and I hope you find it interesting. So as I was introduced, my name is Danyivid Olivares-Villagomez from Vanderbilt University Medical Center. So I don't know the background of many of the people that are listening to this stream. So I will have to give you some information, preliminary information about what I'm going to be talking about. So today's paper is going to be, the title is Osteopontin and I-seeded alpha cells promote intestinal intraepithelial lymphocyte homeostasis. This was, as I mentioned, published last year in the Journal of Immunology. This is by my group. And mainly this work was done by Ali Nazmi, a postdoctoral candidate. He's been in my lab for around 3 to 4 years. And he's just got a faculty position at the Ohio State University. So he's living and also there are other people in my lab, Michael, Kristen, and Blanca that helped in this paper. Anyway, so let's talk about an introduction to the intestines, because this paper is going to deal with intestinal intraepithelial lymphocytes. And the first thing that we need to do is to know where these cells are and what are these cells. So first, this is, if you're familiar with intestines, so here we have the small intestine that is, it starts in the at the end of the stomach and then ends in the colon. And then we have the colon, we have the ascending colon, the transversal colon, and then the descending colon. If we think about the intestines in a very simple way, we can think about them as a tube, right? So we make in this diagram that I'm showing here on the right, if we make a transversal cut in the intestine, it's basically like a tube, right? It has a hole in the middle, and then it has the epithelium surrounding it. So the problem with this is that whatever is inside of the intestines, and this we call the lumen of these intestines, is sort of a representation in some manner of what is outside. Why? Because just think about it, the intestines of the gastrointestinal tract is an open system in which, well, we ingest food in the mouth, and then goes to the stomach, and then to the intestine, and then it is expelled, right? This is an open environment in which we put food particles inside, and there are probably pathogens that come with those food particles that get into what we call the lumen of the intestine. And so we have all this food that we ingest. We have pathogens that are present, but also we have a collection of microorganisms that are the commensal organisms, or the microbiota, that are within the lumen of the intestine. Now, this is all good because, well, we need to get energy from somewhere, right? And the food provides our energy, and we absorb the nutrients through the intestine. But this is also problematic in the sense that for the immune system, this is a very heavy antigenic load because there are so many things that are traveling through the intestines. And on top of that, if you think about the intestines, come back to this slide. If you think about the intestines, if we put them, if we straighten them up, and then we flatten the surface of the intestine. So as you can see in this figure, the intestines have a lot of folding. So if we flatten those foldings out, it is thought that the surface, total surface of the human intestine is around the size of a tennis court. And as you can imagine, that's a lot of surface. And it's a lot of surface that is interacting with all these antigenic stimuli that are present in the lumen. And the immune system needs to negotiate with this intestinal content and determine whether it is going to respond to some of these antigens or is going to be tolerant to some of these antigens. So let's dive in a little bit into more about the structure of the intestines. And I'm going to use this cartoon that was generated by Hilda Charutri in a review in Nature Reviews on Immunology. Here you can see that the intestines are basically composed of a monolayer of intestinal epithelial cells that are going to form a barrier between what we call the lumen, which will be this part, and what we call the sterile basolateral layer of the intestine, basically the inside of the host. Now, as you can see, there are a lot of cells that are in this cartoon. The intestinal epithelial cells are here in blue. But I want to draw your attention to cells of the immune system that are present in between the intestinal epithelial cells, and you can see them better here in this zoom out. You can see that they are lymphocytes that are basically squeezing in between the intestinal epithelial cells, and they live in a very intimate contact association with intestinal epithelial cells. These lymphocytes are called intraepithelial lymphocytes. Why? Because they live in between the intestinal epithelial cells. It is thought that in humans, they're surrounding the small intestine, one intraepithelial lymphocyte per every 10 to 20 intestinal epithelial cells. Now, if you think about the normal surface of the intestines, there's a lot of intestinal epithelial cells there, and therefore there are a lot of intraepithelial lymphocytes, or IEL. And that's one of the reasons why a lot of people consider the IEL population to be the most numerous or the largest immune population in the body. And I think I agree with that. The problem with the intraepithelial lymphocytes is that it's not very well studied. We know a good amount of data about the intraepithelial lymphocyte, but not as much as we know about other lymphocytes in other immune compartments. So let me tell you more about the IELs, the intraepithelial lymphocyte compartment. This is not a homogeneous population of lymphocytes. It's on the contrary, it's a very diverse population of cells. And it's kind of difficult to divide them, to classify them, because there are a lot of features, a lot of diverse ways of development, and a lot of diverse function. But what I found is that the easiest way to classify the intraepithelial lymphocytes is by the expression or the lack thereof of T cell receptor in the surface. So we can have T cell receptor positive IELs, and we can have T cell receptor negative IELs. Now, in the T cell receptor positive IELs, we have conventional T cells, those T cells that we know a lot about, like CD4 T cells or CD8 T cells, right? So we have CD4, CD8. Now, the interesting part is that these conventional CD4 and CD8 T cells, they come from the thymus, right? As any other CD4 or CD8 T cells do. They go to the periphery, they get activated by antigens, and some of them, they will migrate into the IEL compartment, and they will reside in the intestinal epithelium, becoming IELs. But what is interesting is that some of those cells will now express what we call the CD8 alpha-alpha homodimer. So CD4 T cells will become CD4 positive, CD8 alpha-alpha, and CD8 T cells will become CD8 alpha-beta, CD8 alpha-alpha positive cells. In addition to these conventional cells, and that turn into these kind of unconventional lymphocytes, we also have a significant fraction of gamma delta T cells that can also express CD8 alpha-alpha. And we have also an interesting population of IELs that are called the TCR alpha beta. They express a T cell receptor, but they do not express a CD4 or a CD8 alpha beta co-receptor, but they express the CD8 alpha homodimer. Now, if we move to the TCR negative IEL population, and this is a less known population of cells, we have innate lymphocytes. We have the LC3s and ILC1-like cells present in the IEL compartment, but also we have this recently described family of TCR negative IELs, which is characterized by the expression of intracellular CD3. They don't express CD3 in the surface, it's just intracellular. And this population can be divided into those that express CD8 alpha-alpha and those that do not. And this TCR negative IEL population that expressed CD8 alpha-alpha was discovered and initially characterized by my group, and we named them as innate CD8 alpha-alpha IELs.
Danyivid, I have a question from the previous slide.
Yes.
Do we know the function of CD8 alpha homodimers?
Yes, we do know. We kind of know what the function is. Okay, so as I mentioned before, the IELs are in intimate contact with the intestinal epithelial cells, right? Mm-hmm. Now, the intestinal epithelial cells in mice, we don't know for sure in humans, they express an MHC class 1b molecule, which is called the thymus leukemia antigen, or TL. TL binds with high affinity to CD8 alpha-alpha. And what we have shown, because we developed the TL knockout animal, is that the IEL compartment in the TL deficient animals is more activated. They proliferate more, they have more cytotoxicity. So what we believe is happening is that the IELs, because they are in the border between the intestines and the lumen of the intestines, they are the kind of the sentinels of the immune system, and they need to be ready to trigger a response towards a pathogen really fast. So they are kind of in this semi-activated state. What TL and CD8 alpha-alpha are doing is to kind of quench the response so that they don't respond too much when it's not needed, right? So that they don't disturb the architecture of the intestine. So basically, the TL CD8 alpha-alpha interaction is an immunoregulatory interaction.
You did mention that some of these lymphocytes, they will come from the periphery. and they would gain the expression of CD8 alpha alpha. So is this gained due to the interaction with TLs or something in the microenvironment of the intestine?
It's something in the microenvironment of the intestine, and there are many factors involved in this. There are cytokines that are important. You have TGF-beta, IL-23, in some minor interferon gamma, that help the expression of CD8 alpha alpha.
Thank you.
Mm-hmm.
And
I have a
sorry. Because I was really curious why those ICD8 alpha and ICD3 positive cells express CD3 only intracellularly. Oh, sorry. Oh my gosh. Why this expression is not on the cell surface?
That's a very good question, and we don't know. They just express CD3 intracellularly, and we have no idea why they do that. So yeah, that's we use it to classify it, right? We realize that they express CD3 and it doesn't move to the surface. Probably doesn't move to the surface because there's not a T cell receptor. But why do they produce CD3? We have no idea.
Thank you.
Mm-hmm. Was there another question?
Yeah, I had one question. Like, what is the origin of these ICD8 alpha cells? Is that because since they do not express the T cell receptor, like, are they not from the thymus? And
No. These cells are not from the thymus. They actually are directly from the bone marrow. And some of the TCR, some of the IELs do come directly from the bone marrow, like TCR, gamma delta cells. Some of them just come directly from the bone marrow as well. Others, they do require the thymus, like the conventional CD4s or the TCR alpha betas. But ICD alpha cells, they come directly from the bone marrow.
So, like, do you think that they migrate to the intestine as like precursor T cells, and in there, they kind of get triggered because of the microenvironment to express the CD8 alpha alpha and form the ICD8 alpha cells?
Yeah, so we believe that the precursors, which still are not identified, they migrate from the bone marrow to the intestinal epithelium, and then they just turn into ICD alpha cells. Now, one of my theories is that ICD3s here in the right and ICD alpha cells are related because they express CD3. I believe ICD3s are the ones that are giving rise to ICD alpha. ICD3s then are the precursors. This has not been, that's my idea. It has not been tested. We are doing right now some single cell RNA sequencing to determine whether that is the case, because then we can determine a lineage development for these cells. All right.
Mm-hmm.
Okay, so let's start with the paper. And the first thing, for those of you that are not immunologists, or that you are just starting to learn about immunologies, this is how we define ICD alpha cells, right? And we use flow cytometry for that. So I'm going to walk through the panel A of the paper, figure 1. And basically what we do is we get an IEL preparation from the intestines, and that is a very easy way to extract the IELs. We just isolate the intestines, we flush the intestinal content, we chop it in little pieces, we put them to shake in a buffer that contains EDTA, and the EDTA is going to allow the intestinal epithelial cells to not be joined anymore, and that's going to release the intestinal epithelial cells as well as the IELs. Then we collect the supernatant, and that supernatant, then we put it in a Perkel gradient to try to get rid of as many intestinal epithelial cells and just recover the IEL population. And that's what we do here. And then we put it in the flow cytometer. So this is what we do. This is a forward scatter and side scatter plot. And we look for the small cells that are a very well-defined population. These are the IEL population. These big cells, most of them are the intestinal epithelial cells, which are just contaminants coming from the extraction, but you cannot get rid of them completely. Anyway, as these are lymphocytes, the IELs express CD45, and then so we gate the live cells that are coming from this dot plot, and then we ask for the expression of CD45, and as you can see, most of the cells are CD45 positive. Then we gate in the CD45 positive, and we can gate the TCR gamma deltas and the TCR beta cells. And as you can see, these two populations represent the largest populations in the IEL compartment. The TCR beta, for example, we can further gate them depending on the expression of CD8 alpha and CD4, and then we have the CD4 positive, some of the CD8 alpha positive cells, and these are the double positive CD4 positive CD8 alpha cells. That's the way that we identify the different population of cells. There are other gatings that we do that I'm not showing here. But what I want to show you is that these tiny population that you can see that does not express a TCR, either gamma delta or alpha beta. These are the TCR negative cells. And as you can see, it's a very small population, very small fraction of cells. It's around 10% of the total IEL population. And then we can gate on these cells, and here in the right in this dot plot, we can gate in based on the expression of CD8 alpha. And as you can see, there's a well-defined population of CD8 alpha cells. And these are the ones that we call I-seeded alpha cells. The rest of the TCR negative IELs will be in this population. All right. So prior to this publication from last year, when we first discovered the I-seeded alpha cells, we realized doing a RNA-seq that I-seeded alpha cells express a variety of things. Among them, well, they express CD8 alpha alpha, which is pretty good. And they express like granzyme B, granzyme A, but they also express SPP1, which is the secreted phosphoprotein 1. And to tell you the truth, when I saw the results of this RNA-seq, this was a few years back, I had no idea what SPP1 was, but these cells express a lot of this. So I had to read a lot about it. It turns out that this gene, SPP1, encodes the protein osteopontin. And as you can see here, I-seeded alpha cells are one of the main producers of osteopontin in the intestinal epithelium when we compare to other cells present in the intestinal epithelium. So what is osteopontin? Osteopontin is a very ubiquitous protein, and it comes from osteo, which is bone, and pontin, which is bridge. And it got this name because it was originally isolated and characterized in the bone matrix. So they call it osteopontin because it's present in the bone, right? And because it's in the matrix, it's probably making a bridge for to connect all the tissue. Osteopontin is a very phosphorylated, heavily phosphorylated protein, and as I mentioned before, it's encoded by the SPP1 gene. Osteopontin is very ubiquitous. It's present in a lot of human tissues and also in a lot of bodily fluids. It's in bone, it's in skin, it's in urine, it's in milk, it's in blood. It's expressed by a lot of different cells, either in the immune system, cells that are not of the immune system. Therefore, it has a lot of pleiotropic physiological processes, which is good because it's a very important protein, but it's also kind of bad when you want to do research for this protein, because since a lot of cells produce this protein, it's very difficult to discern which cells are producing the osteopontin for a specific effect. Now, in terms of immunology, we know that osteopontin promotes the development of Th1 responses, also promotes the maintenance of pathogenic Th17 cells, and is also involved in NK cell homeostasis. And here, this is a cartoon of a Dutch team that they believe that this is the structure of osteopontin in solution. And as you can see, all these, the blue little spheres, those are the phosphorylation sites for osteopontin. All right. So what we decided to do is look into the role of osteopontin in the IEL compartment. And for that, we acquire osteopontin deficient mice. Those are available commercially. It is very interesting that it's such an important protein and is heavily studied in many aspects, but there's not a conditional knockout system for this protein. So the only one, or until now, and I'm going to talk about this further later, but when we started doing this work, there was not a conditional mutant. So we have to work with a global mutant for osteopontin. Anyway, when we obtained the osteopontin deficient animals and we compare the IEL compartment between osteopontin deficient animals and osteopontin competent animals. And what we observe is here is the collection of the different IELs that I mentioned in my original slide, in the initial slide when I talk about IELs. And these are the IELs in the small intestine in the top row, and then we have the IELs in the colon in the bottom row. And as you can see, basically all the TCR positive IELs in the osteopontin deficient animal are reduced, significantly reduced in comparison to the wild type, indicating that osteopontin is doing something to the IEL compartment, especially to the TCR positive IELs. In the small intestine, it appears that the TCR negative IELs are not affected at all by the absence of osteopontin. In the colon, they are. We don't know the significance of this, but it is what we know is that osteopontin deficient animals have a reduced IEL compartment. We look at other immune compartments. We look at the spleen, and we look at TCR gamma delta, TCR beta, CD8 and CD4 positive in the spleen. And what we observe is that the cellularity between the wild type and the osteopontin deficient animals is basically the same. So there's no alteration in the spleens. We also look in the lamina propria, and we look at B cells, we do look at T cells, and there's no alteration in this immune compartment, which suggests that osteopontin deficiency specifically affects the IEL compartment, but not other immune sites. So then we thought, well, probably this is something related to survival of the cells, right? Maybe the cells are not surviving properly in the osteopontin deficient animal, and that's why they're dying. So we did some 7-AAD and annexin 5 stainings, and what we observed, and this is a very busy slide, so I'm just going to tell you what we observed in the population that are in the red square, is that some of these populations, like the TCR gamma deltas or the TCR beta positive, CD4 positive, CD8 alpha alpha positive, or the conventional CD8 T cells, they have an increased expression of annexin 5 in the surface, indicating that these cells are undergoing more apoptosis, so there's less survival. It's interesting that the other population, we didn't observe that. We don't understand why, but it seems to be just specific, the survival for a specific IEL populations, at least in this type of annexin 5 stainings. Now, we also determine whether the cells are proliferating more or less in the absence of osteopontin. If a cell proliferates more, that indicates that it's in a better shape in the IEL compartment and it's surviving better. So we look at a surrogate marker, which is Ki67. And what we observe is that some of the TCR positive population, they are the wild type animals, they have more expression of the Ki67, indicating that these cells are proliferated more in the wild type than in the osteopontin deficient animal, which indicates that there's a better, probably a better survival when the animals are osteopontin competent. All right, so we wanted to do similar experiments in vitro to see whether we can see survival of different IEL populations. So what we did, by flow cytometry, we enrich IEL populations. Then we put them in vitro in the presence of recombinant osteopontin, or also we added anti-CD44. The reason that we added anti-CD44 is because one of the ligands for osteopontin is CD44, and activated cells, T cells, express CD44. And as I mentioned before, IELs are kind of semi-activated, so they do express CD44. So what we wanted to do is we wanted just to block osteopontin from binding to CD44. And then we determined IEL survival as well. So what we did is we cultured different purified IEL populations, and then we added either nothing or recombinant osteopontin or recombinant osteopontin plus anti-CD44. And what I want you to see, and I'm going to focus in the TCR beta positive CD4 positive IELs, is that first, IELs don't like to be cultured in vitro because they die really rapidly. 60% of the cells survive after just 24 hours of incubation in vitro with nothing. But when we add recombinant osteopontin, the survival, as you can see in the gray bars, increases or is maintained. But this can be blunted when we add anti-CD44. And you can see that also as well in other population of IELs. But the response of other population of IELs it varies, because probably the requirements for each individual IEL population is different. But the bottom line is that by adding recombinant osteopontin, the cells, the IEL survive better.
I have a question.
Yeah, go ahead.
Would you think CD44 is one of the receptors that receives the osteopontin?
Yes.
So what do you know, what is the intracellular signaling machinery inside CD44? Because I never looked at it that way.
Yeah, there's some signaling that can, I'm trying to remember right now, but there's a signaling machinery that can stimulate cells to survive. And I'm just right now not remembering exactly what is that machinery, but yeah, it is described. Okay. Sorry that I'm not remembering that right now. All right, so we did similar experiment with the spleen cells. Either we took total spleen cells or TCR beta positive CD44 positive T cells from the spleen. And also we took spleen cells from SPP deficient animals, and we did similar experiments. Bottom line is that in the absence of presence of osteopontin, the spleen cells, they just behave the same. So there's no effect whatsoever of osteopontin in the cells coming from the spleen. It seems that it's only an effect in the IEL compartment. We also studied IELs from humans, and these are neonate IELs that are taken from neonates. These are babies that are born, some of them prematurely, and they need to have the intestines, part of the intestine, to be resected, and we obtain those tissues. So these are not really normal control. These are babies that have some sort of disease, but this is the tissue that we were able to collect. Anyway, what we isolated IELs from the babies, and as you can see, the symbols that are black, different incubation periods. These are cells that receive nothing, it's just media alone. When we add osteopontin, and these are the gray symbols, and each different symbol represents an individual baby. Then when we add osteopontin, then there's an increase in survival. But when we add anti-CD44, you can see that the survival is blunted. We did the same experiment, but this is with adult human PBMC. We obviously cannot get a lot of blood from babies, and we couldn't get blood from the same babies that we got the intestines. So we have to use PBMCs from adults. We cultured them in the same conditions, and what we observe is that osteopontin has no effect on peripheral lymphocytes. So this recapitulates what we see in mice. Humans' IELs are also responsive to osteopontin. All right, so now we wanted to test whether osteopontin is important for migration. For this, what we are going to do is we are going to use an adoptive transfer system. And I'm going to have to explain something before we go there. So if we have peripheral T cells, some of these T cells are going to migrate, for example, to the mesenteric lymph nodes, and they are going to get activated there. And they get activated because of the antigens of the microbiota. And this is in a mouse that is a naive mouse, right? So they're going to get activated. And some of these T cells, as I mentioned before, they are going to migrate into the intestinal epithelium, and they're going to become IELs. And these are going to be your conventional IELs, right? Your TCR CD4 positive and TCR alpha beta CD8 positive cell. This is what happens normally in a mouse. Now you can replicate this by using a RAG2 animal. A RAG2 animal is an animal that cannot produce because of the RAG2 deficiency, which is important for the recombination events that takes in the thymus to generate T and B T cells, and also B cells in the bone marrow. RAG2 knockouts cannot develop T cells and B cells, so they are immunodeficient in that sense. So what you can do is inject T cells from a wild-type mouse into a RAG animal, and these T cells will migrate into the mesenteric lymph nodes, and they will then get activated and migrate into the IEL compartment, and you can track all these activity. So you can also inject these total T cells, in our case, to double mutant mice, which are osteopontin deficient and RAG2 deficient, so they don't have osteopontin in the recipient mice. So we wanted to know whether the T cells that will go into the mesenteric lymph nodes and then migrate into the IEL compartment, whether they will do it properly if osteopontin is deficient in the recipients. And what we observe at 7 days after adoptive transfer is that when we look at CD8s and CD4s, basically there's no difference between the animals. There's the similar migration patterns, so osteopontin is not important for the migration into the IEL compartment. However, we now want to look what happens at 28 days after the migration of the cells into the IEL compartment, whether the cells are proliferating and surviving. Now, for this, I have to mention one thing. We are transferring total T cells. And the reason for transferring total T cells is the following. some people do this kind of adoptive transfer cells by transferring just naive T cells. Excuse me. If we transfer just naive T cells, what happens is that these naive T cells are going to be activated in the mesenteric lymph nodes, and they are going to migrate into the IEL compartment and into the intestine. Naive T cells do not contain regulatory T cells. So if you transfer just naive T cells, they are going to get activated. And in the absence of regulatory T cells, then there's going to be an inflammatory process that is going to happen in the intestines. So in order to avoid that inflammatory process, we transfer total T cells, meaning these cells will contain your naive cells as well as regulatory T cells, which we think they are going to stop the inflammatory process. And this is something that has been studied many, many, many times by many people. So we did that. We transferred the cells into RAG knockouts and SPP RAG knockouts. But now we look at 28 days post-transfer. What we observe is that the CD4 T cells and the CD8 T cells and the CD4 positive, CD8 alpha positive, because remember, some of the conventional CD4 T cells are going to turn CD8 alpha expression, so they are going to turn into the double positive cells. What we observe is that the CD4s, either single positive or double positive, the reconstitution of these cells is lower in the osteopontin-deficient animal, which recapitulates what we have seen in the osteopontin-deficient mouse that is not in the RAG2 background. The interesting part, and this was very surprising, is that when we look at the intestines of the double mutant recipient mice, the intestines were significantly inflamed in comparison to the control animals, because in the control animals, the regulatory T cells are working and are preventing inflammation. You can see here in panel C, this is the weight change. You can see that the RAG knockouts, they maintain their weight. But when the mice are starting to develop intestinal inflammation, they start to lose weight. And you can see the double knockout mice that are losing weight here. When we look at the colons and we score the pathology, we see that there's a significantly more pathology in the double deficient animals. So we were very surprised by these findings because we were not expecting to see disease. We were just wanted to know whether the cells reconstitute better or not, and whether they proliferate better or not in the absence of osteopontin. But turns out that in the absence of osteopontin, the animals are getting sick. So the usual suspects for this were regulatory T cells. So we said probably osteopontin is important for the function of regulatory T cells. So we enrich regulatory T cells, which are FOXP3 positive, and we have the mice that are the red fluorescent protein that are tagging the FOXP3 positive T cells. We incubate them in vitro in the presence of recombinant osteopontin or anti-CD44, as we did before. And then we look at the expression of FOXP3 by RFP analysis. And what we observe basically is that these are the cells that are recovered from this culture. These are the RFP positive cells. In just media alone, it's around 13%. If we add osteopontin, then the percentage of cells that express RFP and therefore FOXP3 increases. But when we add CD44, anti-CD44, then the RFP frequency of cells is decreased, as you can see here in the summary in this graph. So what we are thinking is that osteopontin in the intestines is helping regulatory T cells to maintain FOXP3 expression and thus regulatory activity. Then what we did, it's a similar experiment, but instead of doing in vitro, we did it in vivo, in which we transfer regulatory FOXP3 positive T cells into rat knockout animals or into osteopontin rat double knockout mice. And what we observed, and I'm just going to show you panel E, is that there's more cells that express FOXP3 in mice that are osteopontin competent in comparison to those mice that are osteopontin deficient, indicating that yes, in the absence of osteopontin, regulatory T cells are losing expression of FOXP3. Okay, so I talk a lot about osteopontin and the IEL compartment, but what about I-seeded alpha cells? As I mentioned in the beginning, I-seeded alpha cells are one of the cells that express a lot of osteopontin in the IEL compartment. So we wanted to determine whether I-seeded alpha cells are important for IEL survival. The problem with I-seeded alpha cells is that there's not a knockout mouse that specifically eliminates I-seeded alpha cells. So we haven't found a transcription factor, for example, that is just specific for I-seeded alpha cells. There are a lot of transcription factors or regulatory elements in the genome that if you knock out them, you are not only knocking out I-seeded alpha cells, you're knocking out other IELs population, so that's not good. So we have to do experiments in vitro to try to determine this, the function of I-seeded alpha cells. So what we did is we took osteopontin competent either I-seeded alpha cells, TCR alpha beta IELs, or TCR gamma delta IELs. And we cultured these cells individually in the presence of IELs that are derived from osteopontin deficient animal, and we put them in vitro, and then we measured survival. So we're gonna mention first TCR beta cells as a source of osteopontin, right? So these are these cells. And then we culture this TCR alpha beta with different IEL populations that cannot express osteopontin. This is kind of a busy slide. And what I want you to see is that we are measuring the increased survival in the presence of this particular IEL population that is expressing osteopontin, in this case, TCR beta cells. As you can see, if anything is in the positive, in the y-axis, that means that it's increased survival. So in terms of TCR beta cells, it appears that they can promote the survival of gamma delta cells, but cannot promote the survival of any other IEL that we tested. Now, if we look at the source of osteopontin, now they are the gamma delta cells, then we observe that none of the, well, most of the IEL population that we tested, there's no increased survival in the presence of TCR gamma delta cells that can produce osteopontin. There's a little survival here in the CD8 alpha beta positive, but it's similar survival to what we observe when we add TCR gamma delta plus anti-osteopontin. So we think that this survival that we see here is not a specific for, or is not osteopontin specific. Anyway, when we look at the source of osteopontin being I-seeded alpha cells, as you can see now, all our graphs are in the positive range, meaning that the survival of all these different IEL population increased in the presence of I-seeded alpha cells. But not only that, when we add anti-osteopontin to these cultures, we see that the survival of the different IEL population is blunted. This suggests that at least in vitro, and we speculate that this is happening as well in vivo, that I-seeded alpha cells produce osteopontin, and this osteopontin then helps in the survival of the different IEL populations that we study. Now, one question is, well, are your cells that are the cells that producing osteopontin surviving similarly in your in vitro culture system. And yes, they are surviving similarly by an exit 5 staining. And what we observe is that ICD alpha cells in this in vitro system are the ones that are producing osteopontin. And this is just by detecting osteopontin in the supernatants. So I think this is my last results slide. So what, oh no, I have one more, sorry. We wanted to exclude, because it is known that activated intestinal epithelial cells can produce osteopontin. We wanted to know whether if we eliminate osteopontin from intestinal epithelial cells, whether the IEL compartment is going to be affected. As I mentioned in the beginning, when we started this project, there was not an osteopontin conditional mutant system, which was surprising because osteopontin is very well studied. So we had to develop our own osteopontin conditional mutant. So we developed osteopontin flux mice. We crossed them with mice that express the Cre recombinase under the control of the Villen promoter. So that is specific for intestinal epithelial cells. So we are eliminating osteopontin expression here in the Villen Cre plus minus mice. And these are our control mice. And what we observe is that most of the IEL population, with the exception of the CD4 positive, CD8 alpha positives, are not affected when we eliminate osteopontin expression from the intestinal epithelial cells. So all these results led us to believe that ICD alpha cells may be one of the most important cells in terms of producing osteopontin in the intestinal epithelium, and does the following. ICD alpha cells produce osteopontin, and osteopontin interacts with CD44 in the IELs, and this promotes survival. Now, there's another publication that we didn't talk today, but osteopontin can also promote the survival of ILCs in the intestinal epithelium, or by doing this in a specific models of intestinal inflammation, these ILCs can become pathogenic. So osteopontin can have dual roles. Osteopontin can also support or sustain the expression of OXP3 and therefore promoting the function of regulatory T cells. We believe this is an effect that is also mediated by TL, as I mentioned before, that is expressed by intestinal epithelial cells to modulate the response of ICD alpha cells. And I didn't talk about this this is another publication that we have. ICD alpha cells can express granzymes, and we believe that they secrete granzyme, and they, by doing that, they can increase pathogenicity. But anyway, this is the model that we are working on. We believe ICD alpha cells have other functions. We already have published many of the functions in our initial characterization of these cells. And that's what I have. And
yeah, I have a question here.
yeah.
So for you said that there are other cells that are also producing osteopontin, but the osteopontin coming from these ICD alpha cells has the most impact on homeostasis, right?
Yeah, at least in vitro, that's what we saw, right?
So,
because we know that alpha, beta, and gamma delta IELs can produce osteopontin as well.
okay.
But in our in vitro model, it seems that they do not secrete osteopontin. They may produce it, but they don't secrete it. And it is known that osteopontin can be intracellular or it can be extracellular.
Okay. Is it possible that there is some, let's say, a second signal that is also required for this osteopontin to work, and that second signal is only presented by the ICD8 alphas?
That's a very interesting question. It's possible. We don't know. but that's a possibility. Yeah.
Okay.
So just let me acknowledge the people that work, right? So Ali is the one that did most of the work with ICD alpha cells, and as I mentioned before, he is moving to have his own lab in the Ohio State University. And Kristen, Michael, Kathleen, and Stephen, they collaborate in this paper. I have a bunch of collaborators all over the world, and this is my funding, and that's it. That's all I have.
Thanks a lot, Danyivid, for this wonderful paper. I'll keep it open to questions. I think Vijay has something.
Yeah, thanks a lot for the presentation. It was pretty interesting. I have a couple of questions. In one of the slides, you had mentioned that you had the chance to look at the intestines of prenatal children, I suppose. I was just wondering, like if there was a comparative study, like if the IEL population was a difference once they had actually come to the once they have been out of the incubation and gone home, probably to like a healthy child or something. Is there a difference because since they were born prenatal, and do you think it stabilizes over time to match with the normal children, the IEL populations as well?
So in our initial publication, and this is from 2014, this is an immunity paper in which we describe these cells. We actually had, we were lucky enough to have a collaboration in which we could get intestinal sections from babies that are undergoing necrotizing enterocolitis, which is a disease of mostly premature babies. And we have also the opportunity to get tissue from controlled babies. They didn't have neck, right? They didn't have a massive inflammation of the intestines, but they had some issues. What we observed is that ICD alpha cells are increased in the babies that are undergoing inflammation, suggesting that they may have a role in this process. In the babies that are normal or controlled, ICD alpha cells levels are normal, right? So regarding to your question, so once that the babies, they recover from NEC, the levels of ICD alpha cells, they return to normal. That's what we think it happens.
And the other question I had was, what is the prevalence? Like, did anyone look at if these CD8 alpha cells can be even present in circulation and in very, very minute quantities?
We actually look at a lot of tissues. Where in the first analysis of our cells, of these cells, we look in a lot of tissues. We couldn't find it anywhere.
Okay.
They are only present in the intestine, in the small intestine column, and also in the stomach. They're in the stomach.
And what about the relevance of osteopontin, like in the different tissues? Because it's been produced by many types of cells, so it is probably involved in many other homeostatic functions as well.
Yeah, osteopontin is involved. Name a physiological process, and it's involved in that, basically. It's produced, as I mentioned, by bone cells. It's produced in liver cells. It's produced by macrophages. It's produced by dendritic cells. It's produced by fibroblasts. So it's produced by conventional T cells. So it's very, it has a lot of functions. I mean, it has functions in cancer development. It has function in metastasis. It has function in development. So it has a lot of roles. One of the roles, and I'm going to mention this, I'm going to advertise this. One of the things that we are studying is the role of osteopontin in milk, because milk has a lot of osteopontin. And what we have observed is that mice that were not fed osteopontin in milk, they develop intestinal dysbiosis, meaning that their microbiota is messed up, which is very interesting. So we are trying to figure out what is the role of osteopontin in neonates in the development of the intestinal microbiota.
And there is one more thing is like, once a child is born, like even the mode of birth has like leads to a different kind of microbiota in the intestines. Do you think this also affects, like the microbiota affects what type of cells are present in the, like what type of IELs are present, and the interdependency of the microbiota and these IELs?
Yeah, so we know, just because the anatomical location of the IELs, we know that they are in close contact with the microbiota, right?
Yeah.
Or they can interact with microbiota antigens, or byproducts of the microbiota, metabolites, right? We know that there are specific bacterial species. One is Lactobacillus rhodori, that is required for the development of TCR alpha beta, CD4 positive, CD8 alpha alpha positive IELs. We know that. We know that TCR alpha beta, CD8 alpha alpha IELs also require the microbiota. But there are other cells that do not require the microbiota, like gamma delta T cells, IELs, they do not require the microbiota for development. So it depends on the IEL population. They are microbiota organisms that are help the development of those particular cells. So if you have dysbiosis, like in the case that you mentioned in vaginal birth versus cesarean birth, well, yeah, it's, if the microbiota is different, probably the IEL compartment is going to be different as well, and that may have implications later on in life, right?
Thanks.
I'm very excited about that milk study that you're talking about. She's saying that without osteopontin, there is inflammation in the gut. And I don't know what's your target, but in my head, I feel it has to be something with its ability to keep the Tregs working.
Well, we haven't seen inflammation. What I mentioned is dysbiosis, which means the microbiota is different from mice that had osteopontin in their milk. Now, that can lead to inflammation. And that's one of the things that we want to test, whether the mice that receive milk as pups, whether they are more susceptible to inflammation later on in life, right? Sorry, what was your question?
No, I'm thinking that because Tregs also decide what kind of microbiota you have, because that's the tolerance you have for those, right?
Yeah, that's a possibility. We are trying to study first what the dysbiosis means and what is affecting in terms of the immune compartment. Of course, one of the targets is regulatory T cells, but as I mentioned before, some IELs are affected by microbiota. Other cells that are affected and that can reach the IEL compartment are NKT cells, and they are very affected by the microbiota. So that's one of our target cells as well.
Okay. One thing about Tregs, so I'm very excited about the Treg fightings mostly.
Yeah, they're very interesting.
What do you have any speculation about how osteopontin is working in Tregs? Is it direct effect or indirect?
I think it's direct. Is binding CD44, and CD44 is signaling in the Tregs. What we need to figure out is what is the signaling between CD44 and FOXP3 expression to connect those dots. We don't know, and I don't think it's known how CD44 can enhance expression of FOXP3.
Because I've had a hypothesis. This was actually one of the things that I presented for my oral qualifier, that JAK-STAT signaling that has a big role in maintaining FOXP3 expression. Certain stats are required for keeping FOXP3 intact, and certain stats they do the opposite. So I wonder if there's any JAK-STAT signaling involved. Although just from a quick Google search, I don't see CD44 having any link to the JAK-STAT pathway.
Yeah. So probably via another mechanism that we don't know. And it will be very interesting to know, right? Because of the relevance of regulatory T cells and the relevance of osteopontin, because it's an important molecule, right?
Yeah.
That would be very, very nice to study.
Yeah, definitely it's something one would write a grant about.
Yeah. And we have some preliminary data that we can look at that, right?
Yeah. Okay, guys. Anybody else? So if there are no questions, I think this would be a good time to wrap up this session. Danyivid, thanks a lot for joining us today. It was a great session. I personally learned so much about intraepithelial cells that I would not have had I read 5 other papers. So this was a very meaningful session for me. And everybody watching us, thanks guys. This video will be staying on YouTube and Facebook for later watching. And we'll see you again in 2 weeks. Our next episode is we're going to talk about T cell signaling with a particular molecule called I think SLP76 and more into that part. So yeah, we'll see you again, and bye guys.